In this video, we will discuss about photomorphogenesis. Photomorphogenesis means photo mean light, morpho mean morphology, that is external characters. So how light controls morphology or external characteristics, it is called as photomorphogenesis. So we can define photomorphogenesis. Light may control many key events: growth, development, and differentiation of plant independent of photosynthesis is called photomorphogenesis. During photosynthesis, plants, when they are green in color, in presence of sunlight, they can synthesize their food, and that food can move with the help of vascular tissue from one part of the plant to the another plant, and that will also control the growth and the development. But here we are not talking about photosynthesis. In excluding photosynthesis, how light is controlling growth, development, and differentiation of the plant, this is called as photomorphogenesis. For example, if we grow seeds in the dark, now when seeds they are grown in the dark, they will germinate to form seedling, and such seedlings they are called as dark grown seedlings. And we will see what is the external characters or morphology of these seedlings. Like this is the seed, pea seed, this is germinating, and this is its root system, root formation, and this is the stem. Now stem is quite weak, like this is weak, divided into nodes and internodes, and leaves they are unexpanded, means they're not properly expanded, and they're small in size. And here at the tip we can see it is leading to the formation of apical hook. So seedlings they are pale, they are not green in color, they are elongated, weak, sickly, with unexpanded leaves. But if these seedlings they are exposed to light, what will happen? Now the stem will develop, which is short and thick. So this will develop into nodes and internodes, which are having short distance, sturdy. So seedlings they are sturdy, they are quite thick, green, and they will develop green color. They will develop chlorophyll, short internodes, and leaves they get expanded to manufacture food for the plant, right? So we can see this appearance of the plant where the seedlings they are pale and elongated, and here seedlings they are sturdy, they are green in color, and they have expanded leaves. So we can say light affects the growth and the development. This reverses the etiolation process, because such kind of seedling they are called as etiolated seedling, and this phenomena is also called as etiolation. But when they are properly developed, they have proper growth, development, and differentiation that helps in presence of light. So this is called as photomorphogenesis. So light reverses etiolation, or this is causing photomorphogenesis, that is development, growth, and differentiation. So light also affects many key events, like this can affect plant movements, this can affect growth, this can affect development, this can even affect the differentiation in the tissues. Another important factors which are involved in photomorphogenesis are what is the innate biological control of an organism, what are circadian rhythm, what are biological clocks inside the organism, then what is the temperature in the surrounding, whether it is high or low, and what kind of environmental factors are there, what kind of nutrition is available to the plant, what is pH value in the surroundings. So all these factors will affect the photomorphogenesis. So we know that when a plant is present of sunlight, so supposing this is the plant, this will bend toward the light, right? And this bending of the plant toward the light is also called as phototropism. So sunlight also affect phototropism. Plants they move toward the light. This is called as phototropism. Plants detect the direction, intensity, and wavelength of light, and they will bend toward the light, and photoreceptor in such plant will perceive the light, and these photoreceptor mean receptors which can perceive the light. So these are of two types, phytochromes and cryptochromes. Now, phytochromes they usually absorb red light, and cryptochrome they are photosensory pigment which will absorb the blue light. So there are two kind of reaction. One are called as low energy reaction, and second are called as high energy reactions. So low energy reactions are also called as low irradiance responses. So they will include the lights red and far red light. and they are mediated by the pigment, or we can say photoreceptor, called as phytochrome. Then there are high energy reactions which are controlled by blue and far red light, and these are also called as high irradiance responses. So, if we see the absorption and action spectra of the chlorophylls or plant which are doing photosynthesis, right? So, this is absorption and action spectra reveal that red light and blue light play an important role in the photomorphogenesis. How it is done? We will take the green plant, we will extract its chlorophyll, and we will measure its absorption spectra with the help of a spectrophotometer, and we will find that there are two peaks. This is one peak and this is another peak in case of chlorophyll A molecule. And again in chlorophyll B molecule, this is one peak and this is the another peak. This is the another peak. This is also peak. So maximum absorption occurs in the blue violet part in chlorophyll A as well as in the chlorophyll B. Chlorophyll A absorb at 430 nanometer in the blue violet region, and chlorophyll A absorb in the red region in the 662 nanometer. Now one is chlorophyll is absorbing at which wavelength? So this is maximum in the blue and in the red region of the spectrum. Now if it is absorbing in the blue and the red region of the spectrum, where does photosynthesis occurs more? Again, we will see that photosynthesis will occur more in the blue. This is the blue part. This is showing high value of the action spectra, mean photosynthesis, and this is again the high value of the action spectra, another peak, which is corresponding to the peak of the chlorophyll A and the chlorophyll B molecule. So this will be, development will be more in the blue and red region of the spectrum. So this is the action spectra and this is the absorption spectra of the chlorophylls. So absorption spectra of chlorophyll A and chlorophyll B, action spectra of chlorophyll A and chlorophyll B. So we can say, what is absorption spectrum? Portions of the light spectrum absorbed by chlorophyll solution which appear as dark band, like we discussed here, two peaks are there, constitute the absorption spectrum, and if this is matching with the action spectra also, this is called as action spectra. So the effectiveness of different wavelength of light on the photosynthetic activity of an autotrophic mean which can make its own food is called as action spectrum. In where does action occurs? It occurs in the blue and red region of the spectrum, right? So this will affect the photomorphogenetic effect in the plant. Now we'll discuss certain examples. Like if we take lettuce seeds, its botanical name is Lactusa stiva, and they are germinated, and if we germinate in the dark, they will not germinate, but they will germinate in the red light. So if we are germinating the dark and we expose the seeds to the red light, that is about wavelength of 660 nanometer, this will induce 100% germination. Now we can do this experiment, like these are the seeds which are growing in the dark condition, and here this is showing the germination because this is the seed. First thing which comes out is the radicle, right? So this is showing its germination process, and when it is exposed to red light, we can see 100% germination, and when it is exposed to red and far red light, again this germination is reverse. So maximum occurs in the red light portion. So another effects which are done by the photomorphogenetic effect are synthesis of anthocyanins, right? So we can see here we are doing responses to the red light. Topic is responses to the red light. Like one, red light control the germination in the lettuce seeds, like we discussed. Then another thing is this also controls the formation of anthocyanin pigment which give color to the plant, right? This also affect coloration in tomato, and we know tomatoes when unripe they are green in color, at maturity they turn red in color. So cuticle coloration is controlled in tomato. Then chlorophyll formation, when we expose the plant to the light, in the dark it is yellow in color, but in the light this will become green in color because of the development of the chlorophyll. This also promote protein synthesis in cotyledon and synthesis of ascorbic acid in synapse alba. The experiment was done and it was studied that more protein synthesis occur and ascorbic acid formation is also enhanced. Then red light reverses etiolation, like we discussed earlier, that when dark ground seedlings, they are pale yellow, sickly shaped, they are etiolated. but when they are exposed to light, this will reverse the ethylation process. So this has been studied in the red kidney beans. When they were exposed to light in the dark condition, so ethylation will be reversed. So red light responses, they are governed by pigment phytochrome or photoreceptor phytochrome. So what is a phytochrome basically? This is a pigment protein complex. I mean, this is made up of two things. One is pigment and the second is protein. So this part is also called as chromophore, which is complexed with the protein. So if we see its structure, this is made up of 4 pyrrole rings, which are not in a cyclic form. So this is non-cyclized tetrapyrrolic part. This is called as chromophore. Chromophore is firmly attached to the protein. We can see here, this is 1, this is 2, this is 3, and this is 4. These are 4 rings, 4 we can say hexagonal rings, and these are called as tetrapyrrole. 1 pyrrole, 2, 3, and 4 tetrapyrrole. Because this is not in a cyclic form, so this is non-cyclized, and this is complexed with the protein, so this is phytochrome red, right? Now, if we compare phytochrome far red, this way you will see that here hydrogen is not there. Right? Here hydrogen is not there, and we can again see here at this part hydrogen is not there, right? So there are certain changes which lead to the formation of phytochrome far red, right? So here hydrogen is not there. Here hydrogen is not there. If it is not there, so there is formation of the double bond like this. And here we can see, we can compare. Here it is the double bond, but here there is no double bond because this hydrogen has moved from here to this. So hydrogen changes are taking place. So this is phytochrome far red. So this is red and this is far red, PR, and changes into PFR, and PFR can change into PR. So this is red, this is far red. So PR, when it is exposed to 660 mu micron, this changes to PFR. And when this is exposed to 730 mu micron, this changes to far red light. This changes to PFR, changes to PR. So PFR form of phytochrome is biologically active form, right? PR changes to PFR. So this will initiate various biochemical reactions and photomorphogenetic responses. It also activates various genes on the DNA for production of a large number of messenger RNA for the synthesis of enzyme responsible for carrying out the biochemical reaction associated with the photomorphogenesis. So phytochrome will receive the light, red and far red light, and this will affect the DNA part, and DNA is having genes on it. So genes will be regulated with the help of this red light or far red light. And when genes are regulated, formation of protein is also regulated. So these genes or proteins, they regulate various kind of biochemical reaction, which is leading to this phenomena which is called as photomorphogenesis. So phytochrome controls by interacting with DNA, affecting the transcription process, affecting the protein formation, and this will affect the photomorphogenesis. Another important pigment which is involved in this photomorphogenesis is cryptochrome. And basically, cryptochromes, they are flavoproteins. I mean, they are flavins plus proteins. And they are also called as a photosensory pigment which respond to blue light. So they are blue light photoreceptors. So they usually work in UVA or blue light about 450 nanometer. A lot of work has been done on Arabidopsis thaliana, and these pigments are found in alga, cyanobacteria, ferns, higher plant, even in the animals. So basically they are controlled by 3 genes: Cry1 gene, Cry2 and Cry3. These genes can later on form protein called as Cry1 protein, Cry2 protein and Cry3 protein. Now gene Cry1 is encoded in the nucleus and cytoplasm, and this usually regulates plant growth and development. And Cry3 gene, this is encoded by mitochondria and chloroplast, and usually this is involved in DNA damage repair activity. So cryptochrome is a proteinaceous, I mean this is protein also, this is proteinaceous, having flavins with it, with 2 chromatophores, flavin adenine dinucleotide also called as FAD, pterin 5, and 10 methyl tetra foliate. So they are flavoprotein, flavin plus protein. Usually, cryptochrome also resemble with the photolyase enzyme. Now, what is photolyase enzyme? This is involved in when DNA is exposed to UV light, it is damaged. When it is damaged, then this is repaired with the help of photolyase enzyme. So that is called as DNA repair enzyme. Actually, cryptochrome is not causing the DNA repair, but its structure resemble with the photolyase enzyme, which is involved in the DNA repair phenomena. So some of the cryptochrome mediated responses are this inhibit hypocotyl elongation. Now what is hypocotyl elongation? When a seed germinate, like this is the cotyledon part and this is the plumule part and this is the radical part, right? So this part, this is the root system, and this part of the stem, right, between the root and the cotyledons, right, these are the cotyledon. This is called as hypocotyl, mean below cotyledon, right, and this part is above the cotyledon plumule part. This is called as apicotyl part. So hypocotyl की length जो है that is shortened or that is inhibited with the help of blue light. So inhibition of hypocotyl elongation. This also control stem elongation. This reduces stem elongation. Petiole elongation, because when leaves they become sturdy and they expanded, so this helps in petiole elongation. And this also helps in differentiation of tissues and organs. Like first, when embryo development is taking place, all the cells are of the same type. They have to develop into various organs. Like if we talk about vascular tissue only, they can develop into xylem and phloem. Or if we talk about mesophyll tissue, this will develop into palisade and spongy tissue. So this controls differentiation of tissues and organs. And in some of the plant, this also regulate floral formation or flower bud formation. So this regulate floral initiation, and this also control circadian rhythm. So what is circadian rhythm? Basically, this is a biological clock which is occurring inside the organism. So this is being controlled by Cry1 and Cry2 genes, or Cry1 and Cry2 proteins. And cryptochromes, they are also causing magnetoreception. Magnetoreception is sensing somewhat magnetic field around a particular species, right? So if a plant is growing and this can sense certain magnetic field around it, this is with the help of cryptochrome only. So this is called as magnetoreception. Then phototropism also. Phototropism is, this is the plant, right, and this is the sunlight supposing, and this is bending towards sunlight, right. This is called as phototropism, but now it is discovered that phototropism is controlled by a pigment called as phototropin. Blue light also control opening and closing of the stomata. Usually this induces opening of the stomata. Stomata are, we can say, these are the tiny pores in the leaves which are helping in transpiration, and when they open there is loss of water in the form of vapors from these pores. So when they are open, this is called as stomatal opening, and when they are closed, this is called as closed stomata. This opening is also induced in the with the help of blue light. So we can say cryptochrome, they are found in all plants. They can be found in the animal also. So they are basically having sensitivity to the blue light. So they will respond with the help of blue light, and they also control various kind of photomorphogenetic effect in the plant. They also control anthocyanin production. They control stomatal opening, like we discussed. They control photomorphogenesis. They can control cotyledon expansion, and they can also stimulate many kind of other biological activities inside the plant. So this is all about photomorphogenesis. If you have any question, please ask me in the comment box. If you enjoyed my video, please like, share, and subscribe. Thank you for watching my video.